Sometimes, a small sample of DNA needs to be sequenced or artificially duplicated. This has applications in fields like forensic science or genetic engineering. But this has also raised ethical concerns, as we'll study how Dolly was the first sheep to be cloned back in 1996. Quite often, the sample of DNA that's collected at a crime scene or some dig site might be too small to be studied, so it has to be amplified or copied. So a way that biologists can do that is through a technique called DNA Polymerase Chain Reaction, or PCR for short. Many cold cases have been solved as a result of the DNA PCR. Gary Ridgway, the Green River Killer, was finally put behind bars when a DNA match was found. More or less, the image that's above my head, or the video, is explaining the same thing when it comes to DNA polymerase chain reaction. So the first thing you have to do is you have to heat up your DNA sample so that it denatures, which means it then comes apart. The two strands will separate, and you're then going to cool it down so that you can set in RNA primers. And the RNA primers basically just tell the DNA polymerase in the next step where it's supposed to start copying the sample. So it's a starting point. So you heat it up, you warm it up slightly. DNA polymerase, the blue enzymes that you see there, will then come in, and then it just starts doing its job. Like how we discussed in the previous series, DNA polymerase starts making copies by introducing free bases that are floating in the sample, and then you heat it up again, it denatures, it comes apart, cool it down, add in your RNA primers, and then you tell DNA polymerase to come in, it starts doing its job all over again. So you just repeat this process over and over. It's called heating, annealing, and extending. And after 30 or 40 generations of this, you can produce billions and billions of these copies. So the DNA sample that you create from PCR can then be inserted into a bacteria, because bacteria, they have something called plasmids, which are these circular ring-shaped DNA things, and they incorporate that DNA, and this allows the DNA to be amplified and duplicated even faster as the bacteria multiply, and then you can then collect that sample and then do what you need to do with it. You can sequence the DNA to find out what order the bases are in, or you can just continue amplifying it. We'll talk about DNA sequencing now, which is finding out the sequence of DNA. What order is it in? Is it A, T, C, G, C, T, whatever, okay? And this is important if you want to find out what the sequence of DNA is to match it up to like a paternity test, or you want to match a person's DNA found at a crime scene, so on and so forth. So it's very similar to the process that we've described for bacterial plasmids and then PCR reaction. So you're going to heat it up, you're going to cool it down, so then you bring in your DNA polymerase. DNA polymerase starts adding or extending these bases, these free bases which you prepared beforehand in the solution. So it starts doing its job. The only difference is it will then tack on something known as a terminator base, which we'll discuss later on. But you can see there it has like a hook on the end. So it tells DNA polymerase to stop adding bases, and then you can see that little like red antenna looking thing. We'll discuss that in just a moment. That's a tag where you can identify what your fragment is made of. So you, again, repeat this process. You heat it, you anneal, you extend, you add the terminator base. There's a primer again. Then it starts adding the bases. There's a different terminator base. Now it's the color green. And you have all these different sized fragments. So now you're going to send this thing through something called a capillary tube, which has a slight electrical charge. It's done through something called electrophoresis. These DNA fragments have a slight charge. It's slightly polar. So it gets pulled through. There's this electrostatic attraction, and they get pulled through and they separate by size. So the smallest pieces go in first, followed by the medium-sized pieces, and then you have the longest pieces at the very end. So now it gets read by a laser. So a laser will then read the tag, and now it's the color green. So a recorder or a computer will look at this, and they analyze it by color. So green is like A, purple is C, yellow is G, red is T. So then you look at the colors, and then you can identify what these bases are, and you start to compile a DNA sequence that you can read off. And then you can analyze the DNA sequence, see if it matches up to a killer or someone who's on trial. Enzymes that can splice and slice up DNA come from bacteria. Bacteria cells use restriction enzymes to cut apart viral DNA that invade their cells. So humans have taken advantage of this, and we now use restriction enzymes from bacteria to create samples of DNA that we need to amplify or to study. So we can splice up DNA into tiny little fragments, and we can read them on something known as a restriction map. And then we can then create DNA fingerprints or whole entire genomes where we can match up the DNA sequence of someone's blood sample to someone who is standing trial for murder. Restriction enzymes cut DNA at specific sites. They can actually read the nucleotides, and they know exactly how they're supposed to splice it. And then you have different restriction enzymes that cut at different sites. So a certain restriction enzyme might separate the DNA into 4 fragments, and a different restriction enzyme might cut it into 7 pieces. You then put the DNA fragments through something called gel electrophoresis, which uses an electrical current to separate the DNA based on their sizes. The DNA will migrate to the different ends of this gel because of its polarity, and it's attracted to this electrical current. So after you do the gel electrophoresis, you then produce something known as a restriction map. So these are a pattern of bands on the gel that show the lengths of the fragments, and they are arranged by size. So the smallest pieces will travel the furthest, and then the longest pieces actually stay near the gel. So you could see how that gold, yellow, tan fragment is the smallest one. It's the smallest fragment that was created after the restriction enzymes cut it up, so it traveled the furthest, followed by the purple one, and then you have that orange one, and then the green fragment is the longest one, so then it stays near the top of the gel. DNA fingerprinting is used to identify a person's DNA to a sample that's found at a crime scene, and the chance of having the same DNA fingerprint is a 1 chance in 5.4 million. So now when I say fingerprinting, it's not intended to be used in a literal sense, like actual fingerprints on your thumbs and your fingers, but it's more the figurative meaning in that fingerprints are unique to the individual. So we're looking at a few pictures here where there's a restriction map of the defendant's blood, his own blood sample. So the killer's blood taken from his body. And then in the blue box, you have the blood found on the defendant's clothes. And you can see there, it doesn't really match up to the restriction map found in the green box, but rather it matches the restriction map from the victim's blood. So it's oddly suspicious that this guy here has blood on his clothing that doesn't belong to him. It belongs to the victim. So you could use this as evidence of a murder taking place, because what in the world is some other person's blood doing on your clothes? DNA sequencing, PCR, and plasmid technology have allowed us to make advancements in the field of genetic engineering. This has allowed us to make discoveries in things like cloning, making transgenic plants and animals, but it also raised ethical concerns. So above me is a picture of Dolly, the first sheep to be ever cloned back in 1996. And I'll explain the process here. What you're doing is you're going to take a donor cell from a sheep's udder, and that one that I circled in black will be the sheep that you cloned, because you're going to use its DNA. You can see there the donor has a nucleus. And this is an egg cell that's taken from a different female sheep. But what you do with this egg cell is you're going to take the nucleus out. It's called enucleating. So you remove the nucleus so it becomes a blank slate. Then you fuse the two cells together using an electrical shock. And now you have this fused cell. And what happens is it just divides normally. It begins to trigger the process of dividing, cell division like we discussed. And it becomes an embryo. And what you do is you take this embryo and now you implant this into the uterus of a surrogate mother. Okay, I believe it could be a third sheep. I'm not sure, but you put it in any other sheep and it gives birth, and this is the cloned lamb that people later named Dolly. So this lamb is the clone of the sheep that's at the top there, so I put a check mark next to the two. Okay, not to be confused with the egg donor, because you actually remove the nucleus from the egg cell, and the surrogate mother does not contribute any DNA to the clone lamb. With all this DNA technology, you can clone sheep, you can fix bad genes, you can manufacture good ones. So what you can actually do is you can implant a piece of good DNA, let's say like your blood can't produce good hemoglobin. So what you do is you implant a hemoglobin gene into the cell of bone marrow, and then you implant it into your bone marrow, and then the bone marrow naturally starts producing these cells that actually have good hemoglobin. Transgenic plants can now be grown with DNA that allows them to have more resistance to frost, disease, and insects, thereby producing higher yields of the crop. A transgenic pig can be injected with growth hormones that allow it to produce a meatier pork chop. There are environmental and ethical concerns about genetic engineering, as well as a decrease in genetic diversity, which leaves crops vulnerable to new diseases and pests. There are also many fears about all the bad things that can come from genetic engineering, mainly the topic of eugenics, which is picking and choosing only the best genes. Are we headed down a path where we create designer babies with acceptable levels of IQ, athletic skills, skin color, facial features? These are all bad things that can arise as a result of genetic engineering, as society deems what's good and what's bad. Or what if your employer or health insurance company actually had access to your DNA sequence and your whole entire genome, and they can actually make predictions about your health later down the road? This means your employer could choose not to hire you, or they could just dismiss or terminate you because they see a potential health problem coming up and you're just going to be absent most of the time. Or your health insurance company can also raise the rates of your insurance because they know that you're going to have some disease in the future. Scientists completed mapping and sequencing of the human DNA back in 2003. Now they're identifying genes in the sequence. The Human Genome Project will allow us to make many advancements in the field of science and medicine. So for example, gene therapy will allow us to remove bad genes and insert good ones so that we can cure you of some inherited disease. But where do you draw the line between what's considered a good gene and a bad gene, and who makes that decision? And are we playing God in some ways? We might be headed towards some nightmare scenario in which human beings consider only a certain race or attractiveness or intelligence or height or hair color to be acceptable. And this might lead to situations in which we end up cleaning out our gene pool and cloning only the best human beings. Even though this is kind of far-fetched and it's extreme, it's something to think about when it comes to DNA technology in the future. I hope you found this series informative and educational, and I'll see you next time on Win Biology.